Okay, my friends, buckle up. I am going to show you about the DNA test. I've been questioned about how can you really be sure this DNA was, first of all, from the rocks that I took it from, which are body parts. Secondly, does that DNA actually mean that it came from a human? Because it's bacterial DNA, which sounds like that's not from the human, but that's not the case. It is specific to a human. Let me explain to you the chemistry here, and then we'll go from there. Okay, my friends, the DNA work was done by Helix Biolab. Magnificent work. All right, here's the deal with DNA. The difference between human DNA and that of other species includes this: how it varies, the genome size, human mitochondrial DNA, the gene order is the sequence and arrangement of the genes, nucleotide differences, the base pairs, to track the evolutionary biology, the mutation rate, and the non-coding regions. The D loop, this is what we're talking about, the D loop control region differs between species and is key to tracking maternal lineages. Where did that DNA come from? The D loop is the killer. All right, and then the codon usage, because some animals use slightly different codons. But the mitochondrial DNA, this is the mother's side only, has a couple of specific areas that indicate that it was human and what stage of human evolution it was, and mine were Homo sapien. All right, let me just explain to you what a PCR DNA test is. It takes these products of the DNA and amplifies them. If they're weak, they can amplify them and get a signature. Sometimes it's weak, sometimes it's not so weak. But in my case, 2 of the 3 specimens were literally raw blood. Okay, so how do you get blood out of a rock? Well, you take a rock like this, and then you find a bone foramen in the bone, which this was, and then you find a scab that is on top of the vein and the artery, and then you remove the scab, which is blood. These are all mud fossils. They all still have blood in them. I took that scab and moved it off. This is what's called fibrin. It's a clotting fabric, the same thing you have in your body, and it collects where the break in the vein and the artery are, and then the scab fills over and collects in this, the blood cells, and it makes a scab, and then it seals it off. Now, I said, there's a vein and an artery. Can I prove that? Yes, I can, because there they are right there, the black one and the red one. So what did I do? Well, I moved the scab completely off, and there it is, the black one. You see it coming up the black little tube? That little tube ran to wherever it ran to the next bone. And here's the red one, and that's the artery. So there's the vein, and there's the artery. Now that's out of a bone. All right, I think it was out of this one. It was out of one. I got them all. As a matter of fact, I got one in the microscope right now. I just went out this morning. I grabbed one off. I get these. You can't get bone. You can't get a rock without blood. It's not like you can't get blood out of a rock. You can't hardly get a rock without blood. As a matter of fact, you cannot. This is Helix Biolab. They are fabulous. The guy that I worked with, Tom, unbelievably professional, is a certified laboratory holding several key accreditations that affirm its commitment to quality and regulatory compliance. They got all kinds of certifications. CAP accreditation, FDA authorization. Helix Biolab's platform has been granted FDA authority for its whole exome sequencing capabilities. This is going to be big, making a significant milestone in genomic testing and personalized medicine. That's what I'm trying to work into. Now, I know he got assaulted I know he got assaulted after doing the work for me, and he doesn't do this anymore. He won't test this kind of stuff anymore because of the pushback. I'm sure I would love to have my stuff tested, and I have people that will pay for it, and they did contact Tom, and he doesn't want to do that anymore, and I understand why. But it's a disaster for science. Let me show you the kind of blood that I took out of these samples. He tested them, and he said they were excellent quality, and they were literally raw blood. Okay, this is about the DNA tests, and this was one that was tested. This is this lung right here. That lung, actually blood came out of these. I drilled into here to get the sample. Now, when I took it out of here, it was literally like raw red blood. This is just literally bloody. I mean, it was just raw red blood there. I took a sample out of there, sent that off. That was one of them. All right, so that was one sample that was sent off to be DNA tested. And it came back saying that that was excellent quality DNA. It was like raw blood. Now this is the big boy. That's 3 feet long from the back right there to the tip right here. That is the fingernail. All right, take your time and look carefully. This is not a joke. This is serious, serious business. That is the fingernail. When I found this was intact. It was an absolutely perfect, flawless fingertip. However, that means that it's completely sealed in what's called grip skin. Grip skin is a very thick coating of skin, and there's no blood in it. So the only way I could get blood was to smash this piece off and go down inside to get the blood, which I did, and it came out raw blood, basically. Now, again, this is the fingernail. You see that little flick of fingernail tip here? That's still on top, and some of it eroded away around the edge of the fingertip, but they come in flakes. Your fingertips are made in layers, and that's the layer is still up there, but this would have been more fingertip, and it's gone, but the fingertip is still there. There's a lot of layers, and especially in this thing. It's 3 feet long. Now, this right here is the little bone that goes, it's a little pad that sits in between the fingertips so that they don't rock on the bones. This is the vein and artery, and this was literally still has fingerprints on it. When I smashed into there, this is brand new stuff. I didn't touch it, and I was very careful. I used a mask and all that business. I drilled down in here and took out literally raw red blood. Now, so we got this one. Now, when I said I had to smash this off, I did. And what it did, it came off with the fingerprints. These are sweat pores. That's a fingerprint. It's as wide as my thumb. And that's why I said you can't get blood out of them. There's no blood in here. This is keratinized cells. These are just keratinized grip skin. It's on your fingers. This is what's tough, tough stuff. When they find this, they find what they call plagioclase, because there's all these little fibers in there too. But there's almost no blood. So when I popped this off, it just popped off like a scab. And then right down underneath it is saturated with blood. You see what a fingertip is? Look at this. These are what fingertips have in them for blood. It's just totally saturated, absolutely dense with blood. And I broke it off. Say this is the fingernail right here. I broke it off, and right down underneath it's just saturated with blood. I drilled it out of there, put it in the cellophane, and out it goes. Now, they tested the blood from that fingertip right here, which is this gigantic 3 foot long jobber. Then I also had the lung done, which is this one here. This is the size of a normal human lung. And all this was from the flood. You see how flat that is? The reason it's flat is it died laying like that in the mud, in the flat, boiled in the boiling oceans when Venus almost hit Earth, and it caused a great flood. And that's what happened, and it preserved all of this because of the hot, boiling, silicate, salty waters. What it did was the membranes, the fascia, preserved the contents. I called it fascia facilitated fossilization because the fascia preserved it. Now, so we got the lung tested, and that was fabulously, excellent quality, excellent quality. We had the fingertip tested, excellent quality because it was perfect. Now, the other one that was very poor quality was this one. This is the hand. That's a left human hand. Take your hand, put it back. You're going to feel this here. Take your finger and go like that with your hand. Go way back with it, and you're going to have that piece going right down there, that tendon. And you go like this, you're going to feel it. Thump, 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 thump. That's thump, 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 thump. And there's your pads that go around. Now the big pad is there, the little pad is there. This is the cleave between the pads. This is the grip skin here. I showed you the grip skin on the big finger. This is the grip skin on your palm. Same as your fingers, your toes, you know, your feet. You have that heavy-duty grip skin. It's meant to be just used and wear away and go away. And when they solidified, the grip skin is totally different than the basement layer, which is the pad with all the red fluffy stuff. This is like silvery looking, and it's peeling. You see how it's peeling? That's just what they do. That's what grip skin does. And that hand is just flawless. So here's what happened. I found the hand. I found a bunch of knuckles and fingers and and actually toes too. This is the bone ball. All right, that's the knuckle. Coming off here is the tendon that runs down from it, just like that tendon that runs right off of my finger to the right of my finger here. All right, there's a tendon runs down here, allows my finger to do this. That's this tendon. And that little spot right there in the center is where it meets up with the muscle, and it breaks flat as a pancake off. That right there. Now, this right here is literally the muscle that wrapped the bone ball. The way this eroded is just fabulous, because this side I can see everything. I see the muscle, I see the tendon, I see the abrupt transition where the tendon was. I see the tendon on its top. It died laying like this. And on this side is the bone ball is still intact. That bone is over here, and it's underneath all this stuff. And if this was one of my middle fingers, it would have had one of these on here and one of these on here. So it could go this way or that way. If it's one of the end, if it's my end finger, it's going to be this one right here, and have only one on that side. But if I'm in the middle fingers, they go both ways. And that is the bone ball, and that's the red, fleshy, tendony stuff that wraps over. And it was upside down, just like the whole hand was upside down. So this finger was right there. This bone ball is that bone ball right there. Coming up over the top is this tendon. And I mean, it is a heavy-duty tendon. And we just saw the muscle, we saw all that stuff. So that's off the table. It's there. The palm is there. The fingertips are there, which I had CAT scan. Some of the bones were there in this mix. Where that was, I don't know. But it's a bone, and there's the cartilage. That's it, everything preserves perfectly. There's where the nerves go, and there's your ligament, and the bone breaks off here. You see that little white spot? I'm going to put this in a microscope. That right there is bone. You almost never, ever, ever find bone in mud fossils. It's what's called nucleophilic transition. And it comes in and everything is replaced. It's called nucleophilic substitution by nucleophilic invasion. And it goes through the membrane, they invade, and whatever stabilizes. That must have somehow been down in the mud somewhere, I don't know. But that particular spot still has bone, and you can see it quite easily in the microscope. Virtually none of my other ones have bones. There's just no bones. Like this here is a hair follicle from another giant. This is all giant stuff. Everything I have here, well, not everything, because I showed you this. That's the size of a normal human. I get crazy stuff here. I'm sorry, it's just what it is. That is a giant hair follicle. And here, let me show you. I show this stuff over and over, and they just demean me and laugh. And that's what this whole thing was about today. This guy said, you know, everybody's laughing at you. They think you're an idiot. I go and I see them talking about you a lot, but all they say is you're a fool. And, you know, the flat earth people are the only ones that pay any attention to you, which is what destroyed me. Because the flat earth, obviously the earth is not flat. And everybody came on and, oh, Roger found giants. That means the earth is flat. It absolutely destroyed me. Destroyed me. The earth is not flat. Now this is a hair follicle. You see that? That's the erector pilli muscle. That's the hair shaft, and it's got a flat side to it because it's a curly-haired person, is what I'm being told. That's the sebaceous gland. That's the root, and those two dots right there are the hair papillae. That's 2 inches wide. I figured out it's about 66 times bigger than a normal human. If it was human, I don't know if it's a human or not, but 60 times, let's say, a 6-foot human would be 360 feet tall. That's, you know, fairly good size. I have them bigger, and I'm not kidding you. And they come much bigger. And that's what this whole video was about, that I did about gigantic creatures. And this guy said, you know, you're showing some obvious evidence, but everybody thinks you're an idiot because they think you're a flat earth person. Again, totally destroyed everything I did. All right, so this is a rock. I'm going to show you it in detail in a second. That is what the artery blood is, the red blood. This spot right here, I believe, that little dot right there is where the vein was. Now, veins make lichen. They grow lichen where the vein blood was for some reason. And the red blood, it stays quite fluid. The vein blood hardens up. It turns real hard. A lot of times it just turns black. But this is lichen, I believe, growing on what would have been black. Now, there's a step down here. This is literally the basement layer of your, you know, your flesh. It's all of this is membranes above. This is the interstitium. That's the fluid-filled highway, all through here, this gooey stuff. And then there's a membrane under that, and then you get down to the basement, below the basement layer of the membranes, and then you're into the flesh. All right, all of this, I'm going to move this around a little bit so you can see. Well, you're not going to be able to see that well. I'm going to have to hold this up so you can see it. But yeah, I can see everything in this rock. And here's some more blood over here. That's just dried out. And I'll bet you if I put water on there, it would start bringing up some red. I did put a little water over on this spot. And you see how it brought up the red? Red stays fluid almost forever. Well, literally forever. You rehydrate it. I got to show you something. Wait till you see this. All right, this is one of the bones, and that's the only bone, almost the only bone I've ever found in mud fossils. They completely transition, mostly. But that's a bone, and that had scabs coming out of it. This is a chunk of tendon muscle, but it's got the veins and the artery right coming through here. Now, I'm going to show you something that's going to stun you. You see the top right there? Oh, you can't see it. I got to turn it around. Well, you saw the vein and artery. All right, the black one right there, that's the vein. That's the artery. Now, that fed a lot of blood down into here. And this is what's called a layer of a slurpee. I call it slurpee. Small leucine-rich proteins. What does that mean? It's just saturated with gooey blood. And can I show that? Watch this change as soon as I put some water on there. That's why I say red blood rehydrates instantly. And that's already pretty good. Red, watch this. Look at that. I got it in the microscope right now. All right, you see from up here, I'm looking down on it. It's just nothing but red blood. Now, you got to drill down inside it because I touch these things. Yes, obviously, of course. But you drill down inside, and when you take blood out, and it's blood, it's blood. And basically, that's what I sent off is this kind of stuff right here. Because I drill, you know, once you get in here, you drill down and then you take it out. And anatomically, there's just no question. There's just zero question about this. And they destroyed this, academia has, for the last 10 years. And nobody will do any of this testing. We've tried everywhere, nobody will do it. They're afraid of academia. For doing science, for doing their work, they're afraid of academia. And I know why. They did the same thing to me. They did the same thing to Velikovsky. Anybody that comes up with something that can basically prove something, they can destroy you very easily, and they did. And that's what this guy was saying. You know, they're laughing at you. They think you're a joke, and you know everybody's saying, "Oh no, watch that guy. He's an idiot. Who could believe him? Only flat Earth people believe him." And that's why the flat earthers really did destroy my whole thing, because they said, "Oh, there's giants on the Earth. That means the Earth has to be flat." And that was the end of it. I was done. From that day on, it was over. Now, the tragedy is the people that love God are the ones that the academics are using against me to destroy this evidence, to make it seem silly. They're saying, "Oh, the Earth is flat." And I say, "Please, don't do this. Don't do this. Why are you doing this?" I said, "Look from the space station." Then they say, "Oh, there's no space station." Oh, God. There's a firmament you can't get through. Nobody's ever been to Mars. There's no space. There's no this. There's no... That's just nonsense. Stop this. This is what has destroyed my research, by saying those silly things. The space station's there. I know very, very well. "Oh, have you ever been there?" No, I have not. I don't need to go there to understand what I see. And what I see on Mars, I understand. Please. It's destroyed everybody that's tried to do any research. It's this absolute fanatical attack on me for not saying the Earth is flat. I'm not ever going to say that, so don't do that here. That's what destroyed me, and I get bitter about this. I'm going to tell you right now, because this took right off just like that, and then about a week later, when they all said, "Oh, that's all flat Earth people. That's the only people that believe any of this stuff," and that's what happened. And that's what this guy said today from Austria. I hope he re-comments on this one, and I'll pin it. I lost a comment that he sent, but I would like to see him resend it, because he's right. They're attacking me on that basis, that nobody could believe that, because only flat Earth people believe that. Well, no, not only flat Earth people. People that pay attention believe it. All right, this lung right here is where I drilled down in here and took out literal blood and sent it off to be tested. Now, this is the lung right here. Got it in the microscope. I'm going to moisten that up a little bit. When it first came out of the ground, they come out and they're just literally bloody as the mud dries out from around them. The blood just oozes its way out. And this was just saturated with blood. I mean, literally just saturated with blood. And I will show you right now, when I put some blood, some water on here, it will rehydrate. I have it up in the microscope there. Now, let's get started. All right, this is basically the area I took the blood out of. You got to drill down a little bit through the surface, because yes, it's going to be contaminated with I don't know what. But once you get down inside, that's gone. You're into the actual tissue that's down in there. It still has all kind of blood in there. Now, this is literally red blood. I'm going to put a little water on here. I'm going to give it a minute or two to rehydrate itself, and then I'll show you how bloody this stuff gets. Okay, how hard is it to collect DNA? It's very simple. You use the basic precautions, the hairnet, the mask, the gloves. Now, what do you do with it? How do you get the DNA? Well, you have to extract it, first of all, from inside. Now, what do you do with it after that? Well, all you do is you have a swab. You can't touch it, you can't drop it, you can't do anything to it, and it has to be untouched by anything other than what you're doing. And then you get your swab, you put it in here, you break that off, you put the cap back on, and you send it off. And it has certain numbers on it that says this swab represents whatever it is. Now, we have done this and not had any success that anybody would do it for us. And I have a bunch of these tests, and nobody will test it because it's, just won't do it. So we're looking for somebody to do these tests. I'm showing you how it's done. It's very simple. That's ready to go. It's not contaminated. And I'll show you how easy it is to get blood out of there. All right. This is the lung that I already had tested. Now, that's where I drilled in before. That's not what we're going to look at right now. We're just going to look at this. Now, it's in the microscope. I put my little probe over here. Look at this. This is hard to orient. Right in here, look. It's just blood. The whole area just turns into blood. I'm not even hardly scraping. I'm just barely touching the top. The blood just goes squirting out. You see that? You see, it's everywhere. But you got to get down inside so you don't touch the outside. Now, my stuff was, well, let me show you the test quickly. There's no question it's blood. And there's no question the anatomy is right. And that mitochondrial DNA is specific to Homo sapien, modern human DNA, even though these were giants. And this was not a giant. This was the normal size. But the other ones were giants. All right, look at this carefully. I'm going to vary the light and so forth. So this is red blood. Now, it was moist a minute ago, and you could see the red better. You know what, I'm going to have to put a little more water on there to highlight it. You see, as the water gets in there, it'll turn nice and red. Water makes the blood rehydrate just almost instantly. Now we can see it's a little more red there. Now, as I turn down the light, it becomes more and more apparent. You see how you can see the different contrast of colors now. That, I know what that is, and it's hard to believe, but it's a sweat pore. I'm telling you, this is the basement layer of the skin. This is the same as my belt. You see all these black dots? That's different because that's a sweat pore. Let me get away from the sweat pore. And you see how all of these other ones here, all this black dots and all that little yellow looking stuff, that's strappy fibers that connect to these black dots. This is just, I moisten it up so you can see the contrast in colors. But the black dots are what connect to these strappy things. Hold on. Let me show you the bottom of my belt, this is nothing more than split leather. This is exactly like split leather, and it's the skin of a gigantic human. All right, this is hard to believe, but it's true. This is the back of split leather on my belt. In between all of these little bumps, there's those little tiny fibers, and that's the same as I showed you here. This is basically identical to this, and that is the grip skin from that gigantic fingertip. It still has the fingerprints on it. All right, well, what we're looking at is right there, the back of my belt. Now, I'm going to show you down near where the loops are, where it pulls apart. You can actually see the fibers. But let's get a little closer look up at that. This is genuine split leather cowhide. And this baby's old. I've had this for a bazillion years. All right, so that's what we're looking at is the belt. Now, look at this now. This is the lung. It has the same fabric because a lung has to stretch like a belt, only it's a little stretchier. All right, the belt is the skin. And it's not quite so stretchy. It's flexible. It'll do this and that, but that has to do this. That's a different type of a fabric, but it's still a fabric, just as skin is a fabric. Now, so we were looking down here, which is basically the same as that, which is up here in the microscope that we were looking at. Now, let's look at this carefully. You see all that black stuff? Those are little balls, and they have straps coming out of them. And that's what I'm looking at in my belt here. Now, if I come down this belt to where the loops are, hold on, right there. All right, you see that? Hold on. All right, these are the same, everything's the same, like the little black balls. They're everywhere, and these are the straps that attach to them. You would never see that. You'd never see that. You'd see this kind of a floppy look to it, which is exactly what I was showing you on the lung and on the fingertips. This is heavy duty skin, and it's just like split leather. All right, if you pay attention to amorites, there is no biology in these whatsoever. These cavities were collapsible so that this thing could pull and stretch out. These were the anchors that anchored the tendons long ago, pre-flood. The new version was a ball with a strap. But if you look, not a single one of these has any biology in it whatsoever. These are simply cavities. And as that thing pulled, it would go just like this and collapse so it could stretch out. And they find them stretched out. You see, some of them are just pulled right out. And there's, and as they stretch, they collapse. That's what all of these little curves are in here. If you understand, physics, that thing pulls this way, these things engage into each other. Click, 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 as this thing pulls. It just keeps going on like that. That's the anchor spot. And they see them, they find them like this. Those don't mean much because they're not pretty like that. There's no biology in there whatsoever. These are just cavities. And when it was alive, they were probably sort of fluidish. Crazy as this sounds, there was several different types of skin that had different types of amorites. This one here is kaolin clay, diatomaceous earth. All right, diatomaceous earth, and it is just as fine as can be, but it consists of these creatures. Fossil power. They say this is 400 million years ago or something. No, this was from skin that was on the faces of creatures, the real thin, slippy clay. You put water on it, and it's like slime. That was just like your skin. You take your skin and you scrape your skin, you put water on it, it just feels like slime. Now this is the same stuff, only it's from a coarser area in your body. It's called bentonite clay, sodium manto, food grade, but it's the same stuff. It's these amorites. They're not creatures. Now the nautiluses that's fine, but you try to find yourself any biology in these. There is none, zero. Okay, something that I didn't show you yet, I don't believe, was the CAT scans. And then this is the fingertip And this is 2D, this is 3D. These were done by Jesse Grant and Associates, the best people in the world. Literally. This shows where the fingernail bed was. Alright, the other one, and this is again 2D, and you wouldn't see that just looking at the fingertip. It's almost impossible to see that. Now, the other thing that you can see is that's the bone. You can barely see it. That's the problem with mud fossils. Everything transitions a lot. So this is like clay. And then you hit the bone, it's just a smidge different. And you really don't see that easily. And when they try to find these bones, they actually chip off the flesh to get down to the bone. I'm not kidding you. I've been watching the way they do this. And they take forever chipping around. Let me just show you this fingertip. Hold on. All right, I did show you that the two of them were literally like raw blood, and the third one was from this fingertip, from this hand, and that's the distal phalanges, black bone, and the black bone pattern. That is the vein, still has the tube there. That's the artery. Now, that's that hook. The same as the hook on this one here. You see the hook? Same thing. This one's extremely eroded. This one's hardly eroded at all. But they still have the same hook. And the red blood blows out. You see the red blood blows out, and blows out. These are the three spots on the side of the finger. Artery comes in and feeds all the way around, and then it goes over with capillaries to the vein side. The veins have clamps. They don't allow the blood to blow out, and they pinch right off. That's where the vein, vein, and vein here. And the black pattern here, if you can see it, I think you should be able to see that black pattern, is what's called a distal phalanges. And this is basically the pattern of the bone inside the flesh. Now, what I did was, instead of drilling in, like I did the other ones, I could see that there was red blood in here. Let me put this in a microscope and you'll see. All right, this one here is the fingertip at the very tip that the blood is blown out. That's the skin around it, or the flesh actually. And this is the hole. Now, you see down in here? This is just nothing but red blood. That's just blood. So I went down in here and I scraped down in here somewhere. And that's what I used, probably scraped it from here. But I didn't drill into it like normal. That's why it was so heavily eroded. I probably scratched it right from there. But I decided, well, I'll just take it from without going deep inside and see how well this stuff does preserve. And it preserved well enough to get a positive ID that it was human. There's no question about that. And the negatives controls were negative. It was not contaminated. And it was from this fingertip, from this hand, and the fingers and the knuckles and all that were right there. This happened to be so eroded that I could see right into the artery, and I said, well, I'll just take it right out of there and see what happens. And because he had 3 tests, and 2 of them I knew were going to be good. They were just blood. The third one, well, that's this one, and it came back weak. And I want to make a very special mention about Jesse Grant and Associates. They're in Canada and the United States. They're, as far as I know, the best in the world at this CAT scans. And 2D, 3D, they do virtually everything. And they're a good operation. And they're very, very nice people. Jesse and Fabio, who I dealt with there, were just as nice as they could be. And they did 7 CAT scans for me at no charge, because they knew the importance of this. And academia, once again, fought back. This is a tendon. and that is the blood vessel, artery or vein, I'm not sure which. This is the other tendon, and that's the other vein. I'm pleased this is a vein, I'm not certain. This, I think, is a thumb, and the reason I say that is this is the bone right here. Now, this is that fingertip, and that is the bone. Now, this piece broke off right down to the bone, so you can actually see the bone here. And if you know how to look, whoops, you can't see it. If you know how to understand what you're looking at, the textures and the anatomy, this is what a bone does look like. Now, this is a different material, and you don't see it until you get it wet. It's just like everything else. You got to get a little bit of moisture on there, and then it brings out the colors and the detail. And, you know, if you look at it long enough, you understand that right there is completely different than this, because it's part of the bone, and you would never know it because it's so transitioned, you just wouldn't know. But if you kept banging this off, you'd keep seeing bone appear, these little bumpy spots. That's what bone is like. Right at the end of your bone is where all the little tendons attach in those bumpy spots. All right, I showed you these little bumpy spots down here. That's the same as this right here. This is where the tendons lock in. They're still in here. That's why you see them as bumps. They're little balls. These are boiled out, so you have little cavities. These are still here. That's why you see them in little balls. Up here is where the tendon was. This is it right here. All right, so that was where the tendon was. All right, that's this tendon right here. All right, broken off, we get down to the bone. This side over here is just flesh. And that indicates to me it's a right hand thumb. All right, you take your right hand, you put your thumb out like that, and this side's sort of flat, and that side hangs over a big lump. You see it? You got the same thing here. So you got both tendons coming down, you got both blood vessels coming down, you got the bone, you got the hook which holds onto the tendons, and this piece fell off here, which is part of the tendon, right down to the bone. And that's because this is a 2D. You can see it very well. The 3D is hard to see in the mud fossils because they transition so much. But you can see that's where the bone is. And that's where this piece is broken away, or it was breaking away when I sent it off. And they did a fabulous job, as best you can do. I had other ones done. Well, I had one. I didn't actually have it done. It was somebody else's. It was a head. Jim Birchall and Arlie Caudle found a literal head, and it's fabulous. And they had a CAT scan down at University of Texas, Austin, which was an absolute nightmare. It ended up, well, I'm not going to even talk about it because it was so bad. You would never even believe it. Now, Jesse did a fabulous job, fabulous, and he did 7 different things for me. But again, they were very, very transitioned, so you really can't see a lot in the 3D. The 2D, yes, very, very excellent to see the structure of the tendons, the bone, the different colors which indicate the different bloods. I mean, it's fabulous. 2D is great. And I never thank Jesse and Fabio enough. And they got a pretty good operation here. This is industrial CAT scanning, industrial X-rays, NDT inspection lab, Jesse Grant and Associates. They internally inspect parts and assemblies. They're lab-based industrial CAT scanning, industrial X-ray. They go really deep. Specialized internal part lab pairs certificate-backed services with 2D, 3D, and 3D industrial imaging equipment. They do it all. Archaeology, this is what I want. That's what you people should be getting a hold of Jesse Grant Associates. If you want something CAT scanned and done, I don't know what they charge. They didn't charge me anything because this was 10 years ago. And all that stuff was done. And they were so nice to me. But this is the stuff they do. And they are, as far as I know, they're the best in the world. They do everything. And they've been doing it for as long or longer, I guess, than just about anybody. Certificates to support our non-destructive testing services. ISO 9001, 90, they got all kinds of certificates and awards and everything else. These guys are top shelf. And that's why I always try to go to the top guy. And I did. I went to the top guy in everything. And as soon as academia hit it, they just destroyed it. And the people that wanted to see this stuff investigated and researched and, the discoveries seen were intimidated by academia. I know they were. There's no question whatsoever. And, of course, they tried to intimidate me, but it didn't work. I just kept on doing it and doing it and doing it. I'm going to keep doing it forever. Reality is reality, even though it's a reality nobody expected. Now, just to be certain you understand, there's no question whatsoever. These were humans. Human mitochondrial genetics is the study of the genetics of human mitochondrial DNA. The DNA contained in human mitochondria. Mitochondria is an energy-producing little organelle. And they're inside of cells, and they produce the energy to support the cell. However, they support every single different species with a different type of mitochondrial DNA. So the DNA that's in the mitochondria that's in our cells is from our maternal line, which is MT DNA, mitochondrial DNA. So it's human, and it also indicates what level of the human branch it's on. Human mitochondrial DNA. It actually tells you if it's a Homo sapien or a Neanderthal or whatever it is. And there's different types of DNA, but there's still maternal mitochondrial human DNA. Just a little tweak here, a little tweak here says I'm not actually a Homo sapien. I am a Neanderthal. But I still have the same mechanism that drives the energy production of a human only, not of a monkey or a chimpanzee even, or any of that. We are very close to the chimpanzee, but we don't have the same mitochondrial DNA, no. But 99% or so of the rest is. This is how they test for these different amino acids in the sequences that they're in. Precisely what mine was. Whoops. This here. Now, this is the D loop, and hold on. There's two regions they tested for that if they match, there's no question it's human mitochondrial DNA, which is modern human, and it was. All right, both of the lung and the 36-inch tip, which is a huge, huge fingertip, they were just raw blood. Homo sapien mitochondrial, cytochrome B gene, and D loop region. You look those up and they say, yes, if you got those, you got a human being. And they're Homo sapien. You see, this is basically, they're talking about cattle breeds. It doesn't matter. The mitochondrial DNA, which is this D loop region, is different for every species. And they're talking about 3 native Turkish cattle breeds. They're different cattle, but they have a different DNA signature in the mitochondrial DNA. So they're still cattle. Just like we're still humans, and so were, I guess, the Neanderthal and so forth. But this particular region changes a little of this or a little of that. What it actually changes, I don't know. Maybe you get those heavy eyebrows with the Neanderthals, I don't know. It appears that the mother's side, to me, is the construction side, basically. And the father's side fills in all the gaps. But the mother's side has more to do with the actual physical development of the person. Even though you might be heavily muscular as a male, I still think the mother's side of the DNA has more effect. You know, why do I say that? Because certain types of nationalities have certain characteristics. It's undeniable. Like Japanese, the eyes and so forth. The Chinese have a certain look to them. You know, black people are black. Different colors of brown people with different nose features and all kinds of different things. There's different hair, curly and so forth. These are built in, to my way of thinking, into the mother's side. And then the father's side fills it all out. Yeah, he's going to be big, he's going to be strong, he's going to be whatever, or she's going to be whatever. But the basic construction, I believe, is from the mother's side. Could be from the D-loop region or the other one, but they are two specific female DNA traits. Okay, I'm sure I showed you about the ammonites, and here's a couple of them right here. These are not creatures. There's no biology in here whatsoever. These are empty pockets. And the structure of them allows for springiness. You can pull this, and all of these will collapse into, this will collapse into that, and so forth, and so on, and they just fill in and compress each other. This is actually, can move against this surface. You see in here? This is just dirt. This is silt and stuff that's moved into here. The rest is, there was some kind of crystals in here that were in some kind of fluids that would allow this to crush together. You see that? That fit right into there, exactly perfect. Into that, into that, into that, into that. So what it means is you can pull this and it will stretch and it will have tension. And that's the anchor in the center. And not a single one of them has any creature organisms inside whatsoever. They're just nothing but a blank space. Just like this one here. Hold on. This one you can see. To me, that looks like there was a bloody anchor there. But it's the same thing. It has the same kind of crystals and the same kind of deformation. That would crunch together and this can split away and just, see it? See all the little crinkly stuff here? That's not one big solid thing. That could pull away from it. And they do have them all stretched out. I think I showed you all this before, but I did get interrupted a few times. And now don't forget, this was all pre-flood stuff. This is all died in the Great Flood. These things are gigantic because of the gigantic creatures. And the other things are just crazy because the creatures were crazy. Like the no-toes, spring feet. That's the heel and that's the strap right there. The heel with the strap coming up, the tendon. The heel with the strap coming up. The tibia, the tibia would have sat right here, and that would have come right across and attached to the bone. All right, but of course it's gone. So this is ripped off right here. That's it right there. And then you have an abrupt transition, which gives you a wrinkle zone. And here it is up close. Here it is right there. These are springs in a foot. This is a foot. Of that there is no doubt whatsoever. You see that? That's the heel. That's the little nubbin at the bottom of the heel. You got a really tough spot. That is the strap that comes up all the way up and would have connected to the bone that was coming down here. And it could rock in that cradle. this little wrinkly stuff, you see it? That's nice and flat, but right there it gets wrinkly. You see that? You see that wrinkly stuff? That's called the wrinkle zone. It snaps there, and because it's under tension normally, and you get a wrinkle zone. This loads up this way on its pin and pulls this latch. I studied this thing carefully, very carefully. This here goes this way, and it's spring loaded, and it spins on here, just like the ammonites, only it's in a foot. This has a little latch. You see that? That's a little latch with a wire going all the way up to here. And there's a little wire and a latch up there. If this loads up, it pulls this because it's on a pin. So this has to go that way. What happens? This has to go that way. What happens? The wire has to go that way. What happens? The wire pulls this into this cavity because it's here. It pulls the whole thing around this way. So you load this one up, and at a certain point, this one starts to load up, and you get a double twist, and it pops right back down. And there's the heel, and there's another little latchy thing going on here. I don't know what that's about. All of these colors are colors of blood and, body stuff. And these pins are pins. Now, my foot here is a different style. This is another foot. There's no question about that. But this has, I believe, a single pin, whereas this has two. That has a single pin, and these two springs rock this way against this plate, which it looks like a sole of a foot, but it's not. It's a plate that's built into this. And if you stepped on this, you would wrinkle here, which it does. It would wrinkle up, and as it did, these things would almost like straighten out, but they'd want to be curved back down. So as you stepped, they'd straighten back out all by themselves. You wouldn't have to do anything. There's no... And our tendons work basically the same way. It's just a new improved version. And that's the foot. That right there is the tibia. Not only that, you see right here? The tibia has two little hooks that go over the side. It's a bone right here, but it also has a little thing that hangs over the side. And there they are right there. That's them. And then you get to the flesh. You see those two little hooks right there? That's what keeps the tibia here, so it doesn't slide. It keeps it latched together here because it's always being pulled against the fibula, which is here. And even in these styles, they had the tibia and the fibula. And it's just been an evolutionary process of different types of feet. And I have a bunch of these, not just one or two styles. I have at least 5 different types of feet. And people have found them sending me all kinds of, and I mean, the ones I have are just crazy. Of course, these are just so obvious that there's no denying them. And the other ones, there's no denying either, but you have to, like Tish found a whole batch of these. He just walked out 15 minutes. I gave her a 15-minute challenge. I said, you go out in 15 minutes, you're going to find something. She went out and found these, and this is hers too. Plus, she found a whole leg, and it's got all of these little springs in there instead of tendons. It's very crazy stuff. This is the ones Tish found. She found a whole batch of these within 15 minutes. They're just laying there, basically. And mine is the same thing. They're right on the surface. I think my area here was one of the last areas that they could find refuge at because I'm at the surface. Triassic area is right on the surface of my property. So I would think that that's about as high up as you can get away from the runoff in this particular area, because it's just nothing but body parts, absolutely 100% body parts all over my property. Big ones, small ones, middle-sized ones, crazy looking stuff. A goose, Caesar Augustus, my buddy. He's even got his feathers still, the pattern's still in there. It was stunning, it's stunning. But I am at the top of the Triassic, at the exposed Triassic. So to me, this must be where they all ran to try to get away. Otherwise, you're going to be down there in the mud, literally in the boiling hot mud. And this was where they came to try to survive, and they all eventually died right here on my property. That's all I can take from this. All right, I'm going to be going back at this again. This is the thumbnail I had on here, which was from the POV channel guy. He goes and he finds things on Google Earth, and he goes out and looks at them. And I'm saying the biology is just unbelievable, and the size of things. And of course, I got assaulted by a whole bunch of people. Now, geologists, I asked respectfully, please answer this question. How do you think these formed? And I go, well, all you got to do is read this. Oh, you got to look page 25, or read this, or do, no. You explain to me how that happened. Well, that's just natural cracking and this and that, and it was in the... It's undeniable what I showed. Now, with the evidence I have just shown you today about my mud fossils, there is no question about this. The size of the creatures is beyond anybody's imagination. Nobody suspected this. I understand that. And in order to understand this, you have to be able to open your mind up to say, I need to stop and think. Don't throw away your beliefs. Don't throw away everything they've told you to say and made you smart. Don't throw it away. Compare it. So look what I say and look what they say. And you make up your own decision. All right, I love you all. I know this is dragged on, but I'm going to go back at this again, because there was a couple of comments made. They were valid comments. Here's what I think. Here's what I think, and they were from geologists. But they, and they were being respectful, and I will be respectful back. But I won't be just called an idiot and a crackhead and all this nonsense. These kind of people, I am going to just. They don't belong in the arena of intelligence. They do not belong in the area of investigation. They do not belong in any scientific arena whatsoever. To dismiss the evidence that I have presented without even examining it, and then being arrogant, and then, quite often, they're just bullies, and you can't get past them because we're the peers. We have to review you and allow you to speak, or you don't speak, and that's what's happened to me for 10 years. I am not allowed to publish anywhere except Academia.EDU, which I do have 45 papers up there. Very few people see them, so that's why I turned to YouTube, and I was crushed here too. I have 225,000 subscribers, and I get a couple thousand hits. Is a big deal for me, because they don't announce me, they don't put me out there, and I have been so pushed back at by academia and by people that they're intelligent people. They want to be intelligent, but they have been mentocided. Just look up menticide. It means they've been so heavily indoctrinated, their mind cannot change. Not only that, they are intimidated. Because if they do speak up about this and say, let's take a look at it, they're in trouble. And I'm serious, in deep trouble. They will be looked down upon by all of their friends and peers and family and everything else for being so ridiculous to be able to even consider this. Regardless of the evidence. The evidence has no effect on anything anymore. So that's, I'm just telling you, after 10 years, 15 years it's been. Finally, after about 5 years of just total denial by everybody and being really demeaned, I said, I'll prove this, no question whatsoever. And I did the CAT scans, the specimen, the DNA, the chemistry, the nucleophilic substitution and invasion, how the phospholipids boil down and they become aluminum silicas, which is a feldspar. I went through everything, top to bottom, wrote the paper in 2015, Fascia Facilitated Fossilization, on Academia.EDU. I gave all this information, CAT scans, and everything to Yale. Then they wrote their own paper on, took a credit for it. In 2016, it's called Exceptional preservation of soft-bodied Ediacara Biota. They gave it a name for themselves, which just means creatures. And then they just said, oh, it's only slimy petri dish creatures, no big things like Roger's talking about. I had given them all my evidence. I told them how it happened, and then they took credit for it. That's Yale. Now, I'd like to see Trump take a look at that. I know somebody told me, I found out somewhere that they have about $40 billion, and they're not doing their job. What do you do about that? All I need is one, and I can change education in a matter of a few months to reality, not to just wasted money. Here, give us some more money, we'll keep researching this and that. No, I've shown everything to everybody, and zero responses, because once you come with evidence that makes their world change, and it's the Rogerian argument, where if you come with evidence, you have to try to find a middle ground. My evidence against yours. You're right about this, I'm right about that. They're not right about anything. They're just totally wrong about everything, and they know it, which just makes them not intelligent whatsoever. All they are is they have the ability to repeat what they're told. And then now they're saying it to your kids, and they're charging them thousands and thousands of dollars, putting them in debt for the rest of their life, not doing their job. I think Trump's got to step in on this Doge. Somebody's got to do something about this. I can't do anything. I've been fighting them for years, and I mean fighting. I never withdrew, and I never stood down. And what are they going to do? They can sue me, I imagine. If they think I'm saying something that isn't true, I can prove every single word I said, and everything that I have here can be retested. And Yale did take credit for my work. In 2016, after my paper came out in 2015, and I gave them all my research, I gave it to Derek Briggs at Yale, and he treated me very arrogantly and disrespectfully. And I hope this will come back against him for his fiduciary failure to his students, his abuse of public trust, and his misuse of public funds. Because I know they were funded by NASA to do this study of my work. Because I contacted NASA as well. I said, you got this same stuff on Mars as you have here on Earth. And they ended up going to Yale to fund them to see if this was possible. And they said, yes, but it only happens to slimy creatures. And of course, it only happens, I don't know what they told them about happening on Mars, but it does. There's no question whatsoever. And Bennu, they just came back with, that sample is a literal piece of biology. It is from a heart. I have shown it over and over. It's heart sarcomeres with layers of phospholipid layers. And they said, wow, the phosphate surprise, we got these layers of phosphates. That's the layers of membranes. Phospholipids are membranes. And then they have the little sarcomeres in between. It's just insane that they won't do their job. And that was way over $1 billion that cost us. They won't do their job. They're going to study this for the next 10 years and waste another couple billion. Now they want to go back up to Mars and pick up rocks for another 11 billion when I already know what those rocks are. I know exactly what they are. I don't have to go pick them up. You can see this. There's no question whatsoever what they are. You see, this is what they came back from Bennu with. Bennu is I'll show you what it is. It's a heart. And it's in space. This just happened. Every culture on Earth had the same story about giants and dragons and monsters and battles in the heavens, gods, afterlife. And here is a piece of muscle. That is muscle sarcomeres. And this is the phosphate layers. Layer sarcomeres. Layer sarcomeres. They come up, boom, 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 boom. And this is the actual sarcomeres. They came and they found all the amino acids, the uracil even. They found 16 of the 20 amino acids. They found 16,000 organic chemicals. They found the clay, which is nothing more than eroded flesh and red blood. And this is what muscle looks like right here. This is they're there, the sarcomeres. And they're all over Mars. And that's what I reported to them. I said, here's you got Mars, Mars crab, Mars Morse code, Mars blueberries, the Mars blueberry is identical. Well, let me just show you what they are. But anyway, you see what this is. That's from a heart here on Earth. This is phospholipids, phosphates. Phospholipids, phosphates. These are sarcomeres, little blocky things they pull in like that. And that's exactly what you got here. It's identical. There's no difference whatsoever. The chemistry is the same. The biology is the same. The anatomy is the same. When you look at what it looks like, here it is right here. These are the heart valves. These are the heart valves. And this one here, see that? Look, follow this carefully. See around there? You see that in the middle? These little things? That valve lets blood come this way and not come back. It pinch it off if the blood tried to go back. This one probably going the other direction. And this one I think is the aorta, this big one. Now, not only do we have this one in space, which is the one they just jumped into, and when they hit it, they went way down inside. They didn't realize they were going to go down. They thought this was a big solid surface. No, they went down through the blood and grabbed a hold of the sarcomeres, and came back to Earth with them. Now here's Psyche, which is another gigantic heart in space. Remember the 3 valves? Look at this. Same thing. Same thing. And that's a heart. And that's a tube coming out of the heart. That's not a crater. And that's a tube coming out of the heart as well. There was battles in the heavens. They must have blown each other to smithereens. And I showed you, this is it. These are the hearts. And this is what the hearts look like. This is when they break off. And they all break off right here. Because it's something called iron banding, where you have the blue blood and the red blood, and there's 2 different oxygen states, and they separate. And you end up with this, just like this, and just like that. All right, this is Psyche, gigantic heart in space. And this is what you're looking down into is these valves. The top pops right off. And even the autopsies guys told me that. They said, you know, we never realized why that happened. When they take them apart, they take the heart, you just take it right off. The top comes off, all the plumbing, basically, because it pushes them apart. Iron banding. Now, in space, it doesn't have that effect. Well, it does have that effect, yes. But you don't have, like, there's certain things on Earth where the iron banding will lay this way or lay that way. And the hearts can separate this way or they can separate up and down. I have one here that's separated all like layers like this up and down. And then another guy sent me one that the whole top just popped right off and there was still actually blood in it. I got so much I can go on for days and weeks and months and years, which I have. And I'm not going to stop until this is examined. I'm not asking to be accepted 100%. I'm saying examine it. These Moki marbles right here are the identical to what's on Mars. It was called the Mars blueberries. And those are interstitium balls, which are these. And why are these here? Because they were in skin. The skin eroded into mud. Mud is nothing more than skin and flesh that has eroded. And clay, it's the clay. This is eroding right here. That was skin. And this is the fleshy part below. And inside here, there's all these balls, which are these balls here. These didn't just start growing here for no reason. These balls were in here. They fell down into their own mud, which is their own flesh. All right, and here's what it looks like in the body right here. That's the skin. All right, there's the skin. All this here is all red, fleshy, gooey, floppy stuff so that you can pull your skin this way and that way. And they will all come back to where the balls were attached. The brown parts are strappy little parts. But they do erode much easier. The black don't erode, they just don't erode. The fleshy stuff here just is like Slurpee, that turns into the mud. See this? This is in an electron microscope of a creature's body. I believe it's a human. there's so many layers of tissue that it's just staggering. Look at all these layers of tissue. And that's just in this one little area. And all these balls are everywhere. They just, so that your body can move and do all these things, and they keep it sort of plumped up and able to come back to where it's supposed to be. That's what this is all about. And they end up doing this if they die and erode. And on Mars, you have the exact same thing, and you have the muscle sarcomeres all over the place. You see this? This proves a bunch of things at one time. This is Mars, and don't tell me it's not Mars, it's Devon Island or something. There's no way. This is dusted. You see this? There's no water here. There's never been water rained on this. This has just eroded from dust. These stretch straps attached to the tendon balls, and I believe, well, I know for a fact this is skin, and this is pinched together skin, and this is pinched together skin, and this is sort of bumped up and stretched. This is relaxed skin. It's just sort of being like homogeneous. Here you can see it's stretched. Now, eventually, the dusting off here, as I told you before, these are strappy, they're a little tough. These are not tough. I mean, these are tough as hell. These just don't go bad. They last virtually forever. And that's why you see them just laying around here, because now they're called the Mars blueberries. And it's just dusted off and eroded it down. And this is Mars. Of that, there is zero question. And I have studied this as well as anybody breathing oxygen. Now, this is the Mars Valles Marianus Trench. This is wider than the United States. And the ancient texts say that Venus hit Mars and destroyed it and wiped its atmosphere completely off. No more atmosphere. Which it also removes its magnetic field. The atmosphere is related to the magnetic field. And that's another whole thing. But the magnetic field was, they say it did have a magnetic field. I don't know how they determined that, but it does not have a magnetic field now, is what they say. Again, I'd like to know, I can see the magnetic fields in space with some of our experiments. Now, if they use that same technology that we used and looked at it, maybe they could see. But I don't know if they have or not. But anyway, you've got the trench where everything could have been killed and blown off into space at that time. I don't know. There's the Mars blueberries. That's the same as the Moki marbles. This is the basement layer of skin. Above this would have been skin moving all the time to whatever creature it was. And then inside of the fleshy part were all these balls. Precisely what I showed you right here. And here's the Moki marbles. That's Earth. And here's over in the United Kingdom, and these are the same thing, only they're much bigger. And the moss is growing on these. That's not a stromatolite growing. The moss will eat everything edible, and they'll end up with balls like this. All right, I suppose well, let me just show you this one. This is muscle. That is muscle sarcomeres on Mars. Once again, no erosion. No water erosion, just dust. This is Mars. Now they say continually over many millions of years, layer after layer after layer. No, the thing fell over dead, case closed. A couple hours later, it starts to, dry out basically. In this environment, it just dried out. And here on Earth, they were saturated and soaked for a long time, and that's what caused the mud fossils. Up there, they pretty much eroded, so you end up with stuff like this. And that's just the sarcomeres. That's why you got these lines here and all these little connective tissues. These are tough, they don't erode. That's what makes your muscles work. Your muscle can't just be floppy, gooey stuff. It's got to be gooey where it can pull together, but it's got to have something to pull with. These are the pullers. Those are the tendon. Well, they're the connective tissue. Connect, connect, connect. And in there, they pinch, pinch, pinch. Red is pinch. This is connect. Now, what else can we see? Well, we can see this, which is, what is this, Roger? Well, this is the Mars crab. You see, what the hell is that? That is an artery, and that is a vein. And these are the legs of the artery, which is the blood vessels going in to service the sarcomeres. This was a gigantic muscle. It's right here. You got your vein, your artery, which pumps the blood in, and you got your vein, which brings it back. And that's called the Mars crab. Again, and I reported this. This goes back to 2015, 2014. I reported all of this to NASA about everything I just showed you, the blueberries, the mussel, the crab, the code, the Morse code. They saw all this stuff with curiosity, and then they just, and they still won't talk about it. And then they went ahead and paid Yale to talk about this exceptional preservation, which, yes, it is. But mine is really exceptional, and mine is human, and mine is gigantic. And it was, I gave it all to Yale, and then they wrote their paper on it. All right, I'm going to wrap it up at this. But again, this is up on Mars, and it is on Mars, and this is from NASA. And this is the other thing that annoys the hell out of me. Anything I showed is from NASA. Oh, you can't believe we're on NASA. You're an idiot. You're a fool. NASA's all... And they thumb me down every single time. They thumb me down if I won't say the Earth is flat. It's just insanity. And it really annoys the hell out of me. So don't do it here. I'm done with that. It's over. And that's what's just destroyed my channel from day one. All right, I love you all, but pay attention. Don't just have a mindset that won't allow you to think. Then you become an academic. An academic, this doesn't exist. For them, this just doesn't exist. This is not. I don't care about the DNA. I don't care about the CAT scans. I don't care about the chemistry. I don't care about anything. It just doesn't exist because I'm an academic, and I was told that this is millions of years of layering. And anybody that would think anything else is a fool. And I'm serious. That is exactly their attitude. Flat earthers and academics have the identical same mind. Identical. I don't care what the evidence is. I don't care what the evidence is. I care what the evidence is. I love you all.